Our next speaker, Hae Ock Lee from the Catholic University of Korea, and she's going to talk to you about a lung cancer project. Thank you, Hae Ock.
Okay. Hello, everyone. It's my great pleasure to present the work of our team, single-cell analysis of lung cancer, how we illustrate the cellular and molecular landscape of lung cancer, and how we are trying to take the landscape study into therapeutic translation. My talk consists of 3 parts. The first part is the illustration of the cellular and molecular landscape of lung cancer, especially lung adenocarcinoma. And second part is the characterization of the T-cell immunity using antibody-derived tags and T-cell receptor data in addition to the gene expression data. And the third part is linking the transcriptome data into clinical brain metastasis. Okay, this shows the cellular landscape of lung adenocarcinoma we have generated for about 200,000 cells from 43 patients. We mainly use the paired normal and tumor tissues for the illustration. We also had late-stage biopsies taken from the tissues and lymph node as well. In addition, we had pleural effusion and brain metastasis samples, surgical resection. I think this data set can serve as a great reference for lung cancer study. And the approach that we have taken for the illustration of the cellular landscape is a sequential clustering, the global clustering first approach, and then take each lineage or cluster for the subclustering analysis. And let me just briefly skip through the findings we had. The tumor specific changes are the anti-inflammatory macrophages in lung cancer. It's been shown by other groups as well. And the dendritic cell populations from more conventional types towards the plasmacytoid disease or CD163 positive disease known as tolerogenic disease. Activation of humoral immune responses. So there's a dramatic increase in B cell numbers, both in follicular cell types and plasma cell types. And if we look at some of the patients who had quite elevated levels of B cells, we could find the, let me just move it. Okay. We could find the clonotype that had expression signature for both germinal center genes and also for the IgG positive plasma cells, indicating these cells are differentiated in situ in the tumor tissues and could be tumor specific. The expansion of regulatory CD4 positive cells and CD8 cells with exhausted phenotypes are well-known phenomena in lung cancer and also other cancer types. For the stromal cells, the immune-suppressive and pro-angiogenic signatures for both fibroblast and endothelial cells. For the classification of the cancer cells, we have taken different approaches and the clustering. The extreme inter-patient variability is well-known phenomena for cancer. So to overcome this individual variability, we use the cell ordering algorithm instead of the clustering. We used Monocle 2 here. We could define 3 molecular states for cancer cells. And especially the state 2 cancer, it had a transcriptional program completely deviated from the normal differentiation program. And we could find the signature had a poor prognostic value in the patient bulk transcriptome data. And using the TS2 cell dominance, we could group lung cancer patient as TS2 dominant or other signature dominant. And also this signature can serve as a indicator for drug sensitivity to the ARC-MAP kinase signaling pathways. And using all those information in the subtypes of cellular subtypes, we could generate the map of cellular dynamics in lung adenocarcinoma. And just point of view, the increase of the exhausted phenotype CD8 T cells and monocyte-derived macrophages were seen in early stage tumor and also at the late stage biopsies and metastasis samples. And actually, expectedly, the number of exhausted CD8 T cells or monocyte-derived macrophages were commonly seen for the patients with higher tumor mutational burden, tested by whole exome sequencing here. And after we construct all those different subclusters of cell types, what we wanted to do was to construct the intercellular network map, so we could pinpoint which cell types are tumor specific and which type of interactions are there that we can use as a drug targets or use as a predictive links for the disease progression. So just to show an example, this is inference of molecular interactions between tumor and endothelial cells or macrophages and endothelial cells. And we use the framework of different samples like normal versus tumor or tumor at early stage, advanced stage, or metastatic disease. And we could find some of the molecular interactions that is elevated in the specific samples and between specific cellular types. And those interactions can be, again, used by biologists so they can develop different therapeutic strategies. So this is a brief summary that we did this tedious clustering analysis to find the intercellular and intermolecular interaction network, which reveals the anti-cancer drug targets. And if you think of this approach, you think that it's very important to accurately define the cells subclusters, because this approach will be greatly affected by how you define your cell types. And with the more technology coming in, combining the protein expression data to the gene expression data, we can refine the annotation, cellular annotation for the subclusters, and we apply this to the T cell study. So we use the antibody-derived tag and T cell receptor data in addition to the gene expression data for the depiction of T cells in lung cancer. This would be the first slide you see if you just use the expression data. But we use the antibody-derived tag for CD4 and CD8 for the in silico separation of two cell types. And probably you've seen the paper just released by Raul Satija group. CD4 and CD8 have the most power to define different T cell types compared to other protein markers. So this is the first marker we used for the separation of the T cells. And we have looked at this CD4 positive or CD8 positive cells separately. And using that information, we redefined the T cell types here. The clusters were more clearly defined compared to the gene expression data alone, but the findings were actually similar. We had more precise gene expression data for the clusters, but it was still the upregulation of regulatory T cells in the tumor tissues, and effector-like T cells from the normal tissues, they had the expression of cytotoxic genes. It was CD4 T cells, but it was mostly cytotoxic genes that defined the CD4 cells from the normal lung tissues. And the total number of CD4 cells were comparable, actually much elevated in the tumor tissues, indicating that the tumor tissue microenvironment for T cells, it's more regulated by CD4 cells. For the CD8 T cell clusters, in normal tissues, more effector type T cells could be defined, and they expressed against cytotoxic molecules and also transcription factors like TBX21, EOMs, the T helper 1 type transcription factors. And compared to that, T cells from the tumor tissues, they were in very diverse transitional state. And one of the famous T cell types would be the exhausted phenotype. And interestingly, this cells, actually they express the elevated levels of PD-1, HCR2, the immune checkpoints, but they also express the interferon gamma transcripts. And we think the ones that we define here from those patients are actually more functional T cells with the exhaustion marker expression, not really terminally differentiated cells that's not working at all. Maybe that's why these cells can be targeted by immune checkpoint inhibitors. So we actually kind of reluctant to call it exhausted T cells, even though they had elevated levels of this, because we think it's still functional. Using the T cell receptor information, we find the cluster characterization more. The T cell cluster that had most clonal expansion derived from the normal tissues. And here, the CD8 T cells from tumor, the exhausted T cells, they also had quite clonal expansion. And they also had quite dynamic state transition, especially with the resident memory T cell types. Again, we think this data indicate the TEX we labeled here, they are still in very active state for the functionality. Using the pseudo-time analysis for those CD8 cells supports our interpretation. So the CD8 cells with the exhausted phenotype formed a separate branch together with the resident memory type of cells here. And they had elevated levels of PD-1, HCR2, but they also had elevated levels of interferon gamma. And if we use this pseudo-time information for the patient, we could find these two patients, T09, T36 patient, who had higher level of tumor mutational burden, had elevated numbers of this branch type of cells. So we would keep characterizing these cells more, so we would have other ways to activate these population. And let me spend just a few more minutes for the characterization of tumor genes associated with a brain metastasis. In the previous analysis we compared the tumor cells from the single cell data between those from the primary tumor tissues or from the surgical resection to find the genes that's upregulated in the brain metastasis. And we actually call these genes as brain metastasis specific genes. In another setting, we wanted to find not the brain metastasis specific genes, but those genes which can predict the brain metastasis from the primary tumor tissues. So we collected more samples, actually mostly stage 4 biopsy samples with a clinical annotation. We had 18 patients with concurrent brain metastasis and 30 patients without brain metastasis. And by comparing those two patients, we wanted to find some biomarkers that we can predict brain metastasis and hopefully some ways to prevent the brain metastasis, really drastic results of the lung cancer. So in the clustering analysis, we again use the sequential clustering approach for the global cell type annotation and the subclustering. What we did first was comparing the global cell types, whether there's any gross changes between the brain meta versus non-meta group. Actually, we didn't find much difference for those global cell types. We would keep looking at more refined cellular annotation for any differences. And for the cancer cell signatures, we selected only the epithelial type cancer cells and did the DEG analysis between them. Very straightforward strategy. And by comparing adenocarcinoma or small cell lung cancer, we could find about 49 genes that's more expressed in the samples with brain metastasis or downregulated in the brain metastasis. And using those genes, we checked the publicly available patient-derived xenograft data, whether the ones derived from the primary sites or brain metastasis, they show similar pattern. Not all genes had similar pattern, but some genes had the similar pattern as what we found from DEG analysis. So we pinpoint some of the genes for further analysis. So we are looking at the histology data for the patients with the clinical annotation, and we are trying to set up the metastasis model for the mouse analysis. So I just walked through how we are attacking the single-cell transcriptome data for lung cancer to try to find the points of therapeutic translation. Although I didn't discuss much, there's a tumor heterogeneity. We are trying to collaborate with immunology people for the multiple CAR-T approach, so we can target multiple tumor antigen using the single-cell transcriptome and actually proteome, the site-seq data as well. We could find some known immune checkpoint targets and maybe some new targets. There were this intercellular and intermolecular interaction network that we could pinpoint which would be best to target in more progressed disease or metastatic disease. And there were potentially tumor reactive antibodies and T cells, and it's easier to tackle the reactive antibodies instead of T cells. I wish we have a way to tackle this, use this activated T cells in an easier way, but antibodies are easier right now. So we are generating the antibody to test the tumor specificity. And finally, we are annotating the clinical data for the single cell. So trying to find the brain metastasis specific genes. Since there are other clinical data we can combine, like a response to the immuno-oncology drugs, we would have more biomarkers or targets that we can make the drug response better. The lung cancer study, I've started the study while I was at Samsung Medical Center in collaboration with Eun-Yang Bak at Samsung Genome Institute and Myung-Ju An from the hematology department. Na-Young Kim is a current member of our laboratory, and she's the one who did the most of the primary analysis of the lung cancer. So this is it. Now I can take questions or discuss aspects.
Thank you very much, Hae Ock. So we've already got one question, and apologies for the pronunciation. This is from Kyung Gyal, and lung cancer patients with higher tumor mutation burden had higher percentages of exhausted CD8. Is it the reason or the result of the occurrence of lung cancer?
Well, I think that is not a specific observation for lung cancer. Mm-hmm. So once the cancer with a higher tumor mutational burden tend to have more exhausted T cell phenotype. Again, I'm not sure whether we should call it like a T exhausted, because they might be called T activated as well. And one other example would be the colon cancer with the MSI type. They have number of mutations, and they have quite number of this exhausted T cells. So I would say it's a common phenomenon, but the link for the response to the immune checkpoint is more clear for the lung cancer.
Cool. I've got a couple of questions, and they're sort of more general because I get these questions often. So the numbers of tumor are normal. So how many do you need to answer the sort of questions you're trying to answer? So I noticed one of the, so for example, the brain met study, you had 14 versus 13. I think the earlier one was smaller numbers. So how many do you think you need?
That's a difficult question.
It
You know
is.
that even though we sometimes deceive ourselves, this is a very, there's a lot of number of cells, but it's a very few patients compared to the large cohort studies. So I think it's very important. Even though we have small numbers, we have to just chip in to increase the number and find a way to do combined analysis so we can increase the number of patients or cohorts so we can have more generalized results. So I think it's still a very small number of patients.
And just another one was on the T cell subsetting. So with the site-seq, that was really nice. And I get questions about discriminating T helper cells. I get the impression you need site-seq to be able to do these sort of subsetting. You can't really do it with just transcriptome. Is that your opinion as well?
Well, I like the transcriptome data. It's for some cluster, it's really difficult to tell whether it's a CD4 or CD8, especially for naive type cells. And I don't know, it may not be important to discriminate whether it's CD4 or CD8. So using site-seq data helps, but there's a paper in the either bioRxiv or medRxiv. You use the site-seq data a lot. There are like a different number of markers that helps a lot for the annotation purposes, but some don't. So we cannot use 200 markers. It's just too much of cost. And we also found when we analyzed the site-seq data, we didn't sequence the antibody text enough. So we could not use the information. You have to have certain reads. You have to reach a sequencing saturation point, which is again a lot of cost. So I think you have to select some of the markers that helps the cellular annotation. And I think it's not gonna be 200. It could be like a 10 or 20.
Okay.
So,
thank you. Do any of the other panel members have a question?
I have a quick question here. It's a beautiful talk. So I was wondering actually about the brain metastasis, and there's a lot of interest or increasing number of papers that I see where they're looking at metastasis and looking at metabolic flux. So I was wondering, like in the differential expressed genes with increased metastatic potential, do you see any evidence of metabolism related change, or could you speculate on that?
we actually, some of the genes are metabolically related, but the number of genes are small, so we cannot say anything about the pathways wise. But one gene that we are looking at is actually involved in the metabolism, glycolysis metabolism. So I bet there is a connection.
Yeah, all right, thank you.
Okay, thank you very much, Hae Ock.